And welcome to workshop 5. This is the top 3 abstracts in CFTR. On behalf of my co-moderator, this is Estelle Cormier-Boyaca. My name is Niall McCarty. I'd like to thank you for joining us today. We have 3 really great presentations for you this afternoon. We're going to first hear from Dr. Melis Aksit on whole genome sequencing of individuals with CF reveals unexpected levels of homozygosity encompassing the entire CFTR gene. Then we'll hear from Dr. Kyushik Mun on patient-derived submucosal gland on a chip to study mucociliary clearance. And finally, from Dr. Wei Zhang on CFTR del 508 mutation affects ciliary structure, junction integrity, and autotaxin LPA signaling access in the CF airway. As we go through the presentations, I just want to remind you that on the right side of your screen, or perhaps below the screen, you'll see a text box from Slido with separate tabs for chat and Q&A. We encourage you to chat socially throughout the presentation using the chat tab, but please make sure you submit your questions in the Slido Q&A tab, and then we will queue those up for proposing to the speakers. So just only questions that are entered into the Q&A will actually reach the speakers and us moderators for consideration. So let's go ahead and get started with hearing from Dr. Aksit from the group of Dr. Gary Cutting at Johns Hopkins University. Go ahead.
Thank you very much for the introduction, Dr. McCarty, and thank you for the NACFC organizers for giving me the opportunity to present this project today. It's my pleasure to present this on behalf of the CF Genome Project. I have nothing to disclose. So when we think about genetic variation in CFTR, we oftentimes limit ourselves to the CF causing variant itself. However, each individual harbors genetic variants outside of the CF causing variant, in addition to that variant. For example, here you can see there are 4 individuals, all of which who are homozygous for F508del, but harbor a distinct set of additional variants. Some, as you can see, harbor heterozygous variants, whereas others have homozygous variants. Typically, there is 1 heterozygous variant per 1,000 base pairs. But in some individuals, for example, in individual 4, there will be a stretch of a contiguous length of homozygous genotypes in a single individual. And I'll refer to that as a run of homozygosity. And these runs of homozygosity could be affecting the phenotype. So as a part of the CF Genome Project, we whole genome sequenced about 5,000 individuals with CF, and 2,942 of those individuals had homozygous CF causing variants. To find out some more details about the CF Genome Project, I invite you to look at poster 193, which is being presented right now in session 7, and also poster 202. So here you can see a histogram of the number of individuals who have various lengths of runs of homozygosity spanning CFTR in megabases. And to orient you, CFTR is about 0.2 megabases, and chromosome 7, which is the chromosome that CFTR lies on, is about 150 megabases. So as you can see here, most individuals either do not have a run of homozygosity, so those individuals are not plotted, or they have a run of homozygosity that's shorter than 2 megabases. But by looking at VCF tools and manual review, we identified 73 individuals who had a quite large run of homozygosity spanning a good portion of the chromosome. So there are many possible causes for these runs of homozygosity. One of them is actually, it could be hemizygosity and not homozygosity, which could be confused in sequencing data. So for example, here you can see a pedigree of a trio in which the offspring with CF inherited 1 CFTR allele from her mom and 1 allele from her dad. And the allele she inherited from your dad, as you can see, has a large deletion, which in sequencing data could appear as if it is a stretch of homozygosity. But when you look at the coverage data and the breakpoints, you will see that is in fact a deletion. So In all 73 individuals with a run of homozygosity in our data set, none of them were hemizygosity, so that was not the cause. Another possibility you can see on the right here is 4 unrelated individuals who have very distinct CFTR alleles, but they all share this segment of DNA, as you can see, because it's conserved in the general population. However, in this case, the individuals who even harbored the same CF-causing variants all had a distinct set of homozygous variants. So it was not that the variants were conserved that was resulting in them having a run of homozygosity. Another possible cause could be autozygosity. In other words, homozygosity due to a shared common ancestor. So here, for example, you can see the offspring with CF inherited 2 identical alleles from the grandmother, both through the mother and through the father, because the parents were related. Another possibility is uniparental isodisomy, which normally offspring should inherit 1 allele from mom and 1 allele from dad. But if there is a meiosis non-disjunction error, then both copies could be inherited from the same parent, and that would result in a run of homozygosity. So to be able to distinguish between autozygosity and uniparental isodisomy, we have to look at the genotyping data from the parents as well. So within our data set, there were 9 SIB pairs and 2 singletons with both parents genotyped. And within those individuals, we found that the region of the run of homozygosity in the offspring was always shared with both parents due to relatedness between the parents. So the cause for the ROH in all cases was due to autozygosity in these individuals. In 2 SIB pairs, 1 SIB trio, and 1 singleton, we had genotyping data available for only 1 of the parents. And in all of those cases, we found that the region was shared between the parent type and the offspring, which eliminated uniparental isodisomy from the other parent as a possibility. In addition, given that there were multiple family members with runs of homozygosity, shows that it's unlikely that this is multiple non-disjunction errors resulting in uniparental isodisomy. So we concluded that it is probable autozygosity for these individuals. For the remaining individuals, since we didn't have parental genotyping data available, we tried a different approach to be able to understand the cause for their ROH. For them, we calculated an inbreeding coefficient, which is a measure of the level of shared common ancestry calculated based on homozygosity throughout the whole genome. Individuals with higher inbreeding coefficients have parents that are more closely related to each other. And as you can see in this plot, individuals with higher, larger runs of homozygosity tended to have higher inbreeding coefficients, which indicated to us that the cause for the run of homozygosity in most individuals was likely autozygosity. Next, we sought out to understand which individuals had an ROH and what their genotypes were. So here you can see a list of the homozygous CF-causing variants that existed in the individuals with an ROH, their allele count, frequency, and number of homozygotes in our data set, the number of individuals with a ROH that were homozygous for that genotype in our data set, and the percent of homozygotes with a run of homozygosity. So I'll walk you through a few examples here. F508del was the most common variant in our data set, as it is in the CF population. There were 2,893 F508del homozygotes, and 45 of them, or 1.6% of them, had a run of homozygosity spanning CFTR. In contrast, there were 6 G551D homozygotes in our data set, 1 of which, or in other words, 16.7% of which, had a run of homozygosity. So the percentage is a little bit higher. And going down the list, as we get to more rare variants, for example, there is 1 individual who was homozygous for 711 plus 1 G to T, and that individual did have a run of homozygosity. So as you can see here, the more rare the variant is, they're more likely to exist as a homozygote in the presence of a run of homozygosity. Lastly, we sought out to determine whether this is affecting the phenotypes of individuals with cystic fibrosis. So here on the left, we looked at the sweat chloride levels, and on the right, we looked at the lung function of individuals with and without a ROH. And as you can see, there was no significant differences in the F508del homozygotes. Similarly, we compared cystic fibrosis-related diabetes and chronic PA age of onset in the individuals with and without the ROH that were all F508del homozygotes. And as you can see, there was no significant differences in age of onset of CFRD or chronic PA. So it does not appear to be affecting the severity of cystic fibrosis for the individuals that were F508del homozygotes. So to conclude, there is a common pattern of near identical chromosomal regions, which I refer to as a run of homozygosity, that encompass CFTR in individuals with CF. The runs of homozygosity are due to relatedness between parents in almost all cases. And the rare CF-causing variants mostly occur as a homozygote due to a recent common ancestor between the parents. The severity of CF in F508del homozygotes with and without an extensive ROH showed no significant differences. So with that, I would like to thank everyone who has contributed to this work. Firstly, I'd like to thank Gary Cutting, Scott Blackman, and Elizabeth Pugh for their mentorship and guidance in this project. Also, I'd like to thank Karen Riri, Hua Ling, and Kurt Hetrick for their contributions to the analyses. And also, I'd like to thank the CF Genome Project investigators for making this data set available, the Cystic Fibrosis Foundation for funding this project, and the patients, care providers, and clinic coordinators for making this data set available through the patient registry. And with that, I can take any questions.
Great, thank you, Dr. Axett. Do we have any questions? Maybe I'll start with one. You showed that the non-F508del variants were more likely to exist with runs of homozygosity than the F508del homozygotes. What do you think is the implication of that? You showed that very nice table. What are the implications?
I guess what that really shows us is that these rare variants are more likely to be occurring as a homozygote in the context of a run of homozygosity. So these individuals normally likely wouldn't have gotten CF if it wasn't for this context of a run of homozygosity. So the presence of this run of homozygosity is making them have these rare variants as a homozygote, which normally wouldn't happen if it wasn't for the run of homozygosity. So I think the important implication there is that we're seeing rare variants as a homozygote, which typically doesn't happen.
Got it, got it. Okay, here's a question from Dr. Ineke Brockman. Could you spend some more time explaining the graphs comparing lung function of ROH and no ROH people with CF? And
Yes.
Secondarily, how did you control for different modulator availability?
Yes, that's a great question. So I'm not sure if you can see my screen still,
Yes,
but I've
still can.
I've shared that slide again. Sorry, I went through it very quickly. So here, each data point represents one individual, and the data points on the right are the individuals with the run of homozygosity, and on the left are the individuals without the run of homozygosity. And there were 45 F508del homozygotes that we have on the right here, and the remaining individuals were F508del homozygotes without a run of homozygosity. So hopefully that clarifies the plot. And to answer the other question, how did we correct for modulator use? So the data we used was we filtered individuals that had modulator use and only took data prior to modulator use. So that's how we corrected that for that.
Great, thank you. I'm going to correct myself. That was actually from my grad student, Kirsten Cottrell. So another question, in other diseases, what is the most common reason for ROHs? Is what you're observing in CF unique for the non-geneticists in the group?
Based on the literature I found, ROHs do mostly occur in the context of autozygosity. Uniparental isodisomy is pretty rare. It typically occurs in about 1 in 2,000 individuals, and that's across the whole genome. Chromosome 7 specifically, there was a 23andMe study published last year that showed that chromosome 7 actually doesn't have uniparental isodisomy very frequently. It's quite rare. So, but generally, I guess since you asked about all other diseases, yeah, runs of homozygosity mostly occur in the context of autozygosity, but there are some chromosomes, for example, chromosome 16, I believe, is uniparental isodisomy occurs a little bit more frequently. So depending on where it is in the genome, runs of homozygosity could be more likely to be due to uniparental isodisomy as well.
Okay. If we have time for one more question, this one is from Dr. Brockman. Did you find homozygosity in intragenic polymorphisms? And if so, did they correlate with the ROH you found?
Yes, so we looked at, this was whole genome sequencing. So we didn't just look at the exonic variants, we looked at intronic variants, and we also included variants outside of the gene as well. So the runs of homozygosity that I'm talking about in this data set were over 8 megabases long, whereas CFTR is just 0.2 megabases. So the variants within CFTR, within introns of CFTR and outside of CFTR and non-coding regions were all also homozygous. Okay,
great. Thank you, Dr. Axett. I think we'll move on. Just to remind you, that was poster number 57, if you need to look it up later. Next up, we have Dr. Kyushik Moon from Cincinnati Children's, the lab of Dr. A.P. Naran. Go ahead, all yours. We can't hear you, you're muted still.
Sorry. Hello, everyone. Thank you for giving me this opportunity. Today, I'll be talking about patient-derived. Why is not moving? Today, I'll be talking about patient-derived some mucous gland on a chip to study mucociliary clearance. We have no financial interest for this project. As you know, cystic fibrosis is caused by mutation in CFTR protein, which is chloride and bicarbonate transport channel on the apical membrane of epithelial cells. It plays an important role in maintaining water balance on the apical surface. Defective CFTR function leads to development of thick or sticky mucus that causes cystic fibrosis. It involves multiple organs, including the pancreas, gut, liver, and lung. In the lung, mucus plugging is one of the major issues in CF patients. It causes inflammation, bacterial infection, blocked gas exchanges, and shortness of breath. However, mucus secretion is very important in maintaining healthy lung because not only it washes the bacteria and dead cells away, but also it secretes the antibacterial components. There are two predominant mutations in the lung, MUC5AC, which is produced by airway surface goblet cells, and MUC5B is produced by some mucous gland. In this talk, I will focus on this MUC5B producing some mucous gland and gland duct. They are located under the airway surface layer nearby cartilage. In the human lung bronchus, the white arrows indicate cartilage. From this lung bronchus, I was able to isolate some mucous gland by microdissection. In general, the mucus is produced by this mucous gland and is secreted to the airway surface through this gland duct by propelling action of ciliary cells. We verified MUC5B is highly expressed on this mucous gland. From this mucous gland, we obtained some mucous gland epithelial cells. We plated the cells on a transverse membrane and switched it to air-liquid interface condition once they were confluent. The cells then polarize on the porous membrane within 8 days. The progenitor cells differentiated to ciliary cells and mucus-secreting mucin cells. We also verified the CFTR is located on the apical membrane of epithelial cells, and cytokeratin 14 is highly expressed on these basal cells. From our isolation and cell culture protocol, we were able to reproduce in vitro human lung gland duct structure where ciliary cells are present and MUC5E is highly expressed as we observe in vitro. We also verified MUC5AC is highly expressed on this airway surface from human lung tissue. We monitored CFTR function using a short circuit coronal assay. In this, non-CF, some mucous gland epithelial cells showed good response to the first choline. We also obtained the cells from a CF patient who has double copy of delta 508 that caused the CFTR degradation in the ER that can cause trafficking disorder. From the short circuit coronal assay, we observed the CFTR function was collapsed in this delta 508 some mucous gland epithelial cells. We examined these cells using RNA-seq analysis before and after differentiation. Surprisingly, CFTR transcripts between CF and non-CF cells were almost the same. To rescue this CFTR function, TriCaptor, which is composed of 2 correctors and 1 potentiator, was developed and approved by FDA last year during this conference. I remember a lot of people happy with that. I incubate these cells with a combination of 2 correctors for 48 hours and ran the assay. The CFTR function was robustly improved, as you can see here. By adding the potentiator, we observed another spike of the current change. From this data, we verified the TriCaptor obviously the promote increased the CFTR function. As you can see here, a mucous layer developed on the cell surface. It can be washed away by ciliary movement. We named this the mucociliary clearance. To investigate this mucociliary clearance, we monitor ciliary movement. The cells, the cilia work so hard for our lungs healthy. We compared the ciliary V frequency between CF and non-CF cells. The data on your right panel showed significant difference between these two. However, you will notice that the absolute value of ciliary V frequency were not very different. Actually, when I look under the microscope, they look very similar. Although the ciliary movement was this fast, sometimes I observed mucous layer was not move away. In the video, the mucous layer, which is out of focus, is connected to the strand-like structure, and it's just spinning around, not moving away. While I was monitoring the ciliary movement, the sticky mucus attached to the lens of the microscopy. So I was curious about this mucus, so I controlled the stage down. The sticky mucus was then elongated with over 1 inch long without disruptor or disconnection. Some groups observed the MUC5E expressing the mucus came out through the gland duct to the airway surface while maintaining this strand-like structure. So my hypothesis was if this MUC5E expressing mucus is too sticky in CF patients, it will hold the mucous layer on the airway surface tightly, then it is hard to wash away. To investigate this sticky mucus, we designed and fabricate some mucous gland on a chip through photolithography and soft lithography technique. It is composed of 2 cell culture chambers and separated by a thin layer of porous membrane. It has opened the top chamber for air-liquid interface condition, and also it will help to collect the mucus. Using this some mucous gland on a chip, we have successfully cultured some mucous gland epithelial cells over 2 months, and we were able to reproduce. We were able to observe the mucus secretion, as you can see here. But unfortunately, somehow I haven't seen the ciliary cells yet. But I'm working on the some modification to have a ciliary cells. It will help to study the mucociliary clearance. This chip is the transparent and easily portable, so we can connect to a peristaltic pump for perfusion system. So by controlling the flow rate on the bottom chamber, we can mimic the in vivo physiology. So I expect this system will also help the drug screening or drug discoveries. In summary, I have generated the protocols to isolate and culture patient-derived some mucous gland and some mucous gland epithelial cells, and successfully differentiate the progenitor cells to ciliary cells and MUC5E expressing mucin cells. We developed some mucous gland on a chip to study mucociliary clearance, and also we observed the mucus secretion on the chip. Using this some mucous gland on a chip, I will generate a method to collect the mucus, and will investigate the mucus for composition, viscosity, and pH from different mutations. And also I will investigate the ciliary movement and mucociliary clearance regarding the CFTR function. I would like to thank all the collaborators and especially Dr. Nerons, and thank you for your attention. Thank you.
Thank you, Dr. Moon. Do we have any questions? Here's one. Dr. Moon, this is from Bob Emmelkamp. Dr. Moon, what other test systems on a chip are you developing or have developed?
I developed.
Yeah, what other test systems on a chip have you developed?
So far I developed the pancreas on a chip and gut on a chip, and also the liver on a chip. So far I published the one of paper regarding to the pancreas on a chip.
Okay, great. Namsoon Ju asks sort of two questions. He says, excellent work. Number one, do you expect ciliary movements on your SMG submucosal gland model are unidirectional? And number two, could you elaborate a bit regarding the orientation of the SMG epithelial cells on your chip?
Actually, the ciliary movement is looks like it's depend on the how the surface is the generate the cells. Like sometimes the cell is multiple layer, so sometimes it's flat and sometimes has the more cells. So based on that geometry, the ciliary movement is ciliary. The direction is different. And also, what was another question?
The second question was, could you elaborate a little bit regarding the orientation of the epithelial cells on your chip?
I think I observed in vitro we have the multiple layer of the cells with base cells to the ciliary cells. Maybe using this some mucous gland on a chip also same have a same the structure of the cell line.
Okay. And Horst Fischer has a question for you here. Which cell types of the gland are represented in the gland on a chip model? And I might actually ask you to also address the distribution of different cell types. Are they the same on the model as what you would see in the gland?
Yes. Actually, from the base cells, there is the we already observed by some specific biomarkers. For example, cytokeratin 14 is one of the base cells markers. So it is highly expressed and is differentiated to the ciliary cells and the mucous secreting cells. So that is the in the future study we are monitoring which point is they differentiate over
Okay,
time.
great. Here's one from Eli McDonald. What are the lifetimes of these chips and can they be frozen and stored?
Yeah, I never reused this chip, but yes, it is the I mentioned is portable, so we can set up on the microscope, so we can see the cell function in real time.
Okay, and similar question, how long is the gland on a chip alive? How long are the cells alive?
So far, I have seen over 2 months, but I experienced over more than 2 months. You can keep culturing if you have a good condition.
Okay, great. I think that may be all the questions we have. So thank you again, Dr. Moon. Appreciate your presentation.
Thank you so much.
All right, next up is Wei Zhang from UT, University of Tennessee Health Science Center at Memphis. And you're muted still. There you go.
Okay, it's okay now? Okay, hello everyone. My name is Wei. I'm from Memphis, Tennessee. I have no relationship to disclose regarding this talk. First, I will give a brief introduction of cystic fibrosis. CF is caused by the loss or dysfunction of the CFTR channel function resulting from the mutations. F508del is the most common mutation. Clinically, CF can affect multiple organs. The chronic lung disease is the most cause of morbidity and mortality for CF patients. Shown in figure 2 is a hypothesized pathophysiological cascade of CF lung disease. The dysfunction of a CFTR channel causes the depletion of airway surface liquid, defective mucociliary clearance, mucus obstruction, chronic infection, and inflammation. So this vicious cycle would cause bronchiectasis and respiratory failure. So there are two aims for our study. Aim number 1 was to investigate if and how F508del affects the structure of airway epithelium. So the aim number 2 was to investigate if ATX and LPA2 axis plays a role in the fluid homeostasis that's about the CFTR channel function and the inflammatory response. To study the mucus clearance in CF, we used the tissue, trachea and bronchus from CF patients. We also used tissue from normal subjects as controls. Our data showed a MUC5A was accumulated in CF, trachea, and bronchus, suggesting that the mucus clearance was impaired in CF. To investigate the structure of cilia, we used isolated tubulin as a marker. Our data showed compared to normal tissues, the isolated tubulin was decreased in CF trachea and bronchus. To further investigate how this decrease occurred, we checked two membrane trafficking proteins called RAB5 and RAB11, and our data showed that in CF trachea, the expression level of RAB5 and RAB11 was decreased, suggesting that F508del might cause a defect in ciliary vesicle transport. We also checked the two pathways related to the cilium, the P38 MAPK pathway and the hedgehog pathway. Our data showed the phosphorylated P38 and ERK was increased in CF trachea, suggesting the P38 MAPK pathway was activated. We also found that several effectors in the hedgehog pathway was altered in CF trachea compared to normal. Before I present in data of ATX-LP signaling in CF airway, I will give a brief background information. So we all know that severe neutrophil-dominated inflammation and chronic infection are hallmarks of CF lung disease. So the CF-kappa-B pathway and the calcium mobilization in the airway epithelial cells are very important to control lung inflammation through the regulated production of mediators, including IL-8. IL-8 has been shown to participate in the recruitment and activation of neutrophils, modulation of apoptosis, and control of epithelial barrier integrity. So the elevated ATX expression, LPA production, and their signaling pathways have been reported in multiple lung pathologies, including inflammation, fibrosis, and cancer. And LPA itself has been shown to regulate the migration of a variety of cells, including fibroblasts, epithelial cells, and immune cells. So our hypothesis was in CF lung disease, ATX and LP signaling plays an important role in the regulation of CFTR channel function and the neutrophil-dominated inflammatory response. Among the 6 LPA receptors, LP2 receptor forms a complex with CFTR at the apical plasma membrane of airway and intestinal epithelial cells. The formation of such a complex would inhibit CFTR channel function through the LPA2 mediated GI pathway. So in the CF airway, so this complex could be targeted to attenuate this inhibitory signaling. So 2 approaches can be used. The first one, you can disrupt the interaction between LP2 and NACFC. So the second one, you can also antagonize the LP2 receptor. So in addition to increasing CFTR channel function, these 2 approaches also has a potential to attenuate the LPA2 mediated production of some pro-inflammatory cytokines and chemokines like IL-8. Okay, to investigate the ATX and LP signaling in CF airway, we first tested the expression of ATX and LP receptors. Our data showed ATX and LP receptors, especially LPA1, 2, and 3 are upregulated in CF airways. So shown in panel A is mRNA data, the panel B is Western blotting data, C is immunofluorescence staining data. We also tested the LP expression in CFBE cells. Our data showed in F508del CFBE cells, the LPA2 expression was upregulated compared to wild type and parental cells. To investigate if disruption of a NACFC LP2 interaction potentiate the channel function of F508 CFTR, we used a small molecule called CO068. So this is a specific disruptor for LP2 NACFC interaction. Our data showed compound CO068 augmented the channel function of the rescued F508del CFTR, showing in figure 9 is using chamber data in CFBE cells. DMSO was used as a negative control for compound CO068. Showing in number 10 is data from the mouse intestinal enterosphere swelling assay. So phosphoclean was used as positive controls. DMSO was used as negative controls. So we can say that this compound can augment the wild type CFTR channel function and also the VX809 rescued F508del CFTR channel function. To investigate if this complex regulates IL-8 secretion, so we used 3 approaches. So the first approach is to inhibit LPA2 receptor by using a specific inhibitor called BAC35. So our data showed BAC35 can decrease the basal and the IL-1 beta induced IL-8 secretion. The second approach is to rescue the F508del CFTR by using VX809. Our data showed VX809 can slightly decrease the basal IL-8 level and significantly reduce the IL-1 beta induced IL-8 secretion. So the third approach was to use CO068 to disrupt the LPA2 and the NERV2 interaction. Our data showed LPA 18:1, so actually can induce significant amount of IL-8 secretion in CFB cells, and compound CO068 can decrease the LPA-induced IL-8 secretion. So far, all the data are presented in airway epithelial cells. So how about the ATX LP signaling in neutrophils? 2 types of neutrophils were used in our study. The first one is neutrophils isolated from the human peripheral blood. The other one is neutrophils differentiated from the HL60 cells. So we first tested the expression of ATX and the LP receptor in these neutrophils. Our data showed that LPA1 and LPA2 are the most abundant LP receptor in these neutrophils, showing panel A are mRNA data. Panel B are Western blotting for LP1 and 2 in these neutrophils. Panel C is immunofluorescence staining for LPA2 and LPA1 in human neutrophils. So we next tested if LPA actually induces the migration of neutrophils. Our data showed that LP18:1 could induce the migration of these neutrophils, and activation of LPA receptor using a specific agonist called C143 can also induce the migration of neutrophils. In conclusion, our data showed in CF airway, F508del causes the impairment in ciliary structure and function and the upregulation of pro-inflammatory signaling pathways like P38, MAPK pathway, and the ATX-LP signaling axis. All these factors could work together, contributing to the pathology seen in CF lung disease. In CF airway, so the ATX-LP signaling pathway and this complex could be targeted to augment CFTR channel function, suppress the production of pro-inflammatory cytokine and chemokine production, and 3, potentially could mitigate the excessive infiltration of the neutrophils and the subsequent inflammation. All these effects would be beneficial to CF therapy. Much of this work was done by Drs. Kong, Zhang, and Sai in my lab, and Dr. Moon in AP's lab. I thank you for your time and attention. I can take some questions.
Thank you, Dr. Zhang. We're just about out of time, but we do have a couple of questions that we might try to do real quickly. So Azada Paksa has asked, has the decrease in acetylated tubulin positive cells in CF trachea been demonstrated in other studies and contexts?
I'm not aware of that, but I
Okay.
can double check for that.
Great. Second question, have you examined the expression level of phosphatidic acid phosphatases in CF samples?
No. This is a still ongoing project.
Okay.
No.
I'll ask a question. Have you been investigating cells expressing other CFTR mutations other than F508del, particularly ones from other classes of CFTR mutants?
Not in this project. So this project is all about the F508del and the wild-type CFTR. But for this protein complex, so it could be formed between other CFTR variants and LP2. If these CFTR mutations have intact PDV motif at the C terminal, it potentially can form the complex with LP2 and NACFC.
Okay. So do we know if the outcomes you observed are more due to the loss of functional CFTR or due to the expression of a mutant CFTR? Sort of a loss of function versus gain of function.
I think it's a loss of function.
So in cells that don't express any CFTR, you would expect to see the same thing as the F508del cells.
Possibly, but I have to admit that I'm not that sure.
Okay. Great. Well, it doesn't look like we have any more questions, so I guess we'll just wrap up. And I'd like to thank these three young investigators for their really great presentations. So really wonderfully done. Thank you.
Thank you.
And thanks to the audience for attending.